We are talking about treatment of ovarian cancer, and any treatment is all about killing the cancer cells. So first, I'd like to start off by discussing what are the things that keep cancer cells alive in spite of the drugs that we use, in spite of chemotherapy, and so on. So let's look first at the repair mechanisms that are there in a cell that prevent DNA damage, that even after chemotherapy and other therapy will repair the cells and keep it alive. Because what we need to do is introduce some amount of synthetic lethality, identify some weak point in some cancer cells and exploit that weakness in killing the cells. And that is what targeted therapy is about. So there are really two types of DNA repair mechanisms in cells. There are repair mechanisms that kick in to repair single strand breaks, and they're of three types: base excision repair, nucleotide excision repair, and mismatch repair. And then if a single strand break progresses to a double strand break, then there are other pathways that kick into place to repair double-stranded breaks. And there are two of them. One is the HRR, the homologous recombination repair pathway, which is a bit slow, but which is very efficient and very accurate. And if HRR is not active, then you have NHEJ, non-homologous end joining repair, which is not as accurate and leaves some amount of scarring in the genome. So let us now take a look at all of these repair pathways and how do we exploit the synthetic lethality that comes through them. So as I was explaining earlier, you have an intact DNA, and cancer cells, because they're dividing rapidly and so on, they will have errors during the replication process, and that might manifest as single-stranded breaks. Now, single-stranded breaks typically will be repaired by the base excision repair pathway, and then the cell will be completely viable. But if for some reason you inhibit the base excision repair pathway using PARP inhibitors, then the single strand break will progress to a double strand break. And then normally the double strand break will be repaired by the HRR pathway, and again the cell will be perfectly intact. But if you have BRCA mutations, or you have homologous recombination deficiency through any other means, then the HRR pathway is not active. Then the NHEJ pathway comes into place, and that one is not an accurate pathway, because of which errors will accumulate in the cell and lead to cell death. So what we're really interested in before we prescribe PARP inhibitors is to see, is there HR deficiency in the individual or not? If there is HR deficiency, then giving PARP inhibitors will introduce synthetic lethality and cause cell death. So then the main question is about how do we determine whether there is HR deficiency in an individual or not? And really there are two ways of going about it. We could either try to understand the HRR pathway, the genes that are present in the HRR pathway, and check are there any mutations which will cause the HRR pathway to be deficient. Or we could say that we don't really have a full understanding of the HRR pathway, but if the HRR pathway is not active, if there is HR deficiency, we can look for what is the type of effect that NHEJ or the lack of HRR will cause on the genome, and we can just look for that effect. So to summarize, we can try to establish whether there's HR deficiency using two ways. The way that has been used for the last 15-20 years is to understand the genes in the HR pathway and identify if there are mutations in those. And there are multiple genes in the HR pathway. There's a set of 14 genes which people are really confident about, but in some papers there are as many as 43 genes, and you could look for mutations in those genes, either as a germline mutation or somatic mutation. Or you could say that I'm not really interested in understanding the mutations because I'm not always sure about what is the effect of the mutation. Rather, I will look at the effect that will be shown on the genome if there is HR deficiency. And there are three common types of effects that people have measured over the last 10-12 years, and different combinations of these effects. And so the most commonly known effects are loss of heterozygosity, large-scale transition, telomere allele imbalance, and combinations of them. So I'll show you a pictogram of what these types of losses look like. Loss of heterozygosity, as represented here, is that you expect to have two copies of each allele at every point in the genome. But if in some place one of the copies is missing, then it would be called a loss of heterozygosity. And the second one shows where you also have loss of heterozygosity, But more importantly, the entire telomeric region is missing. So it's an imbalance in the telomeric region. And that has also been shown to be one of the effects of HR deficiency. And you have large-scale transitions, which are many copy number amplifications and deletions occurring all across the genome. And that is also one of the effects of HR deficiency. So people look for these three types of patterns of errors spread across the genome and compute whether there is HR deficiency in a sample or not. But since we're talking about molecular testing, I will next show what do these type of errors or patterns look like when we actually do genomic sequencing. And so when we do genomic sequencing to check for HR deficiency, we really get two types of measurements from all across the genome. So we get two types of estimates. One is to say how much imbalance there is in the two different copies of the genome, and that is shown by the top plot. And the second plot is showing what is the total number of copies that are present at any particular locus. So if both of these are perfectly flat lines, then it means that you have equal number of copies of both of the chromosomes, and you have there is no copy number gain or loss. Everything would look perfectly flat. But if you have any variations from the center line, it means there is unequal number of alleles at this location and there is a loss. So we summarize the two top plots out here, which are based on genome-wise measurements, and compute what is the segments that we're seeing across the entire genome. So here, if you were to assume that the red is one copy of the chromosome and the blue is the other copy of the chromosome, you can see that on chromosome 4, you have an entire loss of one of the chromosomes. Whereas in chromosome 8, you have a copy number gain of one of them. So using the genome-wide measurements that are shown in the top two plots, we compute the segmental variations in the bottom plot. And you can see that in the sample on the left, there are segmental variations in two of the chromosomes. Whereas but overall, it's a reasonably flat line across most of the genome. So it is a sample with low genomic instability. Whereas on the sample on the right-hand side, you can see that all across the genome, you see allelic variations, you see copy number changes and so on. And therefore, you see a lot of segments that the chromosome can be broken up into. So that is a sample with high genomic instability. And here I've shown you measurements using the test that we have deployed in the laboratory, which is using genome-wide targeted sequencing across tens of thousands of locations across the entire genome. But in general, measurement of genomic instability, as long as you're measuring a lot of regions, whether you use targeted sequencing or whole genome sequencing, the patterns will look roughly the same. We also did whole genome sequencing for many samples when validating the assay. And you can see that the same patterns that you see using targeted sequencing, you can see here for chromosome 2, the Q arm is showing a variation, and the same pattern is seen even during whole genome sequencing. So you get similar measures, whichever test you use, as long as the test is covering a large part of the human genome. Now the challenge is, here you can see visually that it looks like a sample with high genomic instability, but how do you derive a cutoff and say that, how do you quantify this genomic instability as a number and say that if a sample has genomic instability above that number, you'd call it positive, and below that number, you'd call it negative. And so for that, we can take a look at what was the approach that the Myriad people used around 6 years ago. They used the approach where they sequenced a lot of samples where the BRCA status was known, and they had a measure of genomic instability. And they used the rule of thumb that if a sample has BRCA deficiency, it is expected to have a high genomic instability. If the BRCA is intact in the sample, most of them will have a low genomic instability. So they used a cutoff of, let's make sure that at most 5% of the BRCA deficient samples have a low score, and that resulted in a Myriad score cutoff of 42. So it's really nothing magical about the 42. It's just that they chose it such that 95% of BRCA mutated samples would have a score greater than that. But you'd also notice that there are quite a few BRCA samples which have BRCA intact, the red samples, which also have a high genomic instability score. So this led to the testing paradigm where you can't, where you want to go beyond BRCA testing. You actually want to test for HRD, and samples with samples which have BRCA mutations most of the time will have a high HRD score. But many samples which don't have BRCA mutations will also have a high HRD score, reflecting the fact that the deficiency in the HR pathway might be in some other gene, it might be a methylation pattern, it could be some other unexplained reason which is also causing deficiency. So really, for any particular sample, we need to do two things. Once we get a sample and we extract the DNA, we need to analyze the BRCA genes and see are there any mutations in the BRCA1 and 2 genes. We also need to analyze the entire genomics, whole genome analysis of the sample and compute genomic instability. And if the sample has either a high genomic instability or it has BRCA mutations, we would call out that sample as HRD positive. So just to summarize, if the sample has mutations, BRCA mutations, or the genomic scar score as defined by our assay is greater than 50, it's called high. In either of those two cases, we would say the HRD status is positive. Only if there are no BRCA mutations and the score is less than 50, the test will give a negative result. And here is just an example of some samples that we had done where we found out mutations, we calculated the genomic scar score, and there are different combinations that are present here. I won't go into the details since we explained it. And I just want to show a summary of 64 samples, clinical samples that we had processed as of last month. And you can see that whatever you expect from Myriad and all of the other studies continues to hold up. Almost all samples which are BRCA positive have a high HRD score. At the same time, of the 64 samples, there were 17 samples which are BRCA positive, around 25%. We can't say whether these are germline mutations or somatic mutations. Our test can't distinguish that. But overall, if you're doing only BRCA testing, let us say you're doing somatic BRCA testing alone, then in this cohort, 25% of them, let us say, would have been BRCA positive. But once we add in the genomic scar score also, then 48% of the samples have genomic scar score greater than 50. So it doubles the number of people who would be eligible for PARP inhibitors based on the latest recommendations. And these two statistics fit in almost exactly, I think, with the clinical trials that had been done, which showed that if you were to use genomic instability measurements, you would roughly double the number of people who would become eligible for treatment as opposed to doing somatic BRCA testing alone. And I want to just give you a quick glimpse of what the reports would look like. Here is a report where you have a mutation and the genomic instability score is high. In this case, because there's a mutation, we would also indicate the gene and the variant. And as I mentioned earlier, we can't really say whether it's a germline or somatic. So if you want to have any discussions with the patient on implications for other family members, then we can always do a germline hereditary cancer test, or we could check for this specific mutation in the blood sample and see whether it is germline or somatic, etc. And here is an example of a case where there was no mutation, but the genomic instability was very high. So the report would look more or less the same. It would still indicate the HRD status is positive, but there is no mutation about which you can give any details. And the report would have all of the necessary references to all of the studies which show what the implications of high HRD are. So with that, I would like to conclude my talk and thank you all for being an attentive audience, and thank you, Dr. Talwar, AstraZeneca, and the organizers for inviting.